once we have finished with the basic part, that is still fertilization in plants, in angiosperms, we are going to now continue with the development of the embryo. So what happens after fertilization? So what we have done is explained is how exactly the zygote is formed, right? That is, among one of the male gamete, one fuses with the egg to form zygote, another fuses with the polar nuclei to form the endosperm, primary endosperm nucleus. That was the end of fertilization. So what further happens is the zygote will continue to grow. Zygote is only the first diploid stage formed. After the haploid stage ends, actually haploid stage is the gametic phase or gametophytic stage, where the pollen grains are formed and the egg is formed. That's a very small stage in angiosperms. That's called gametogenesis or gametophytic stage. But once zygote is formed, the sporophytic phase is regained. Then what happens is the endosperm with primary endosperm nucleus. Yesterday I explained you how exactly these will form the various structures for nourishing the embryo. This the embryo will be pushed into the endosperm to get the nutrients. Right? Now we will talk how exactly the embryo is formed. Before that, what are the fates of other parts in the complete embryo? So yesterday when I gave you a diagram, you know the integuments were there above the ovule. These integuments are going to form the future, the seed coats like testa and tegument. They are the seed coats formed. Then other parts of the flower will fall off. Flower, other parts like I told you, the calyx will be there, corolla will be there. They will now have any importance. They will fall off. So only the embryo part we are concentrating now, because this is going to become the future embryo. So what is that stage? From zygote till the formation of the seed, we call it as embryo development or embryology. So as yesterday I was mentioning, the previous class I was mentioning, this major work has been done by Dr. Panchanan Maheshwari. Then you can refer that book if you get it online, or you can get it from any of the bookstores. That is P. Maheshwari, Embryology in Angiosperms by P. Maheshwari, Panchanan Maheshwari. Now, with that book reference, I am giving little higher versions, because once you learn anything, in short you cannot get anything. You have to learn in depth, so that you will feel, this is something really having importance for our studies, not only studies. Some people have been worked continuously on this. Therefore, you just don't go on the surface headings and all, right? So if at all you will be thinking that, okay, I will concentrate only on the board, or I will concentrate on CET, or only on NEET. Don't do that. Everything in depth you learn. Taking exams is the next thing, but learn the subject properly. Therefore, I am giving the elaborative explanation of this whole of the subject. Okay. So what are the stages in the embryology, embryo development of embryogenesis also we call it as, or embryology. First, this is zygote. So zygote is formed at the end of the development, that is the fertilization. Now the question is, after once the zygote is formed, it is not immediately ready to grow into new plant, right? Grow into new embryo. So it will take some time, right? So what is that it is doing is, it will take some resting phase. We call it the phase of dormancy, right? You might have learnt in first PUC under growth. You would have seen most of the seeds will have dormancy period, very long dormancy period. It is called the general resting period for the embryo or the zygote. It will not immediately start germinating. It will stay just for dormant for some time. So whenever it is dormant, then it will develop small vacuoles. You can see lower end of the zygote develop a small vacuole and it start enlarging. Around the nucleus, you get lot of mitochondria developing and number of plastids start growing. Right? Now, zygote, which is a diploid structure, first undergo two divisions transversely, or we call it as the periclinal division. First, it is undergoing periclinal. Anticlinal is longitudinal division. So periclinal it divides, forming two cells. Upper one is called the terminal cell, lower one is called the basal cell. Right? So terminal cell is going to form the future embryo, and basal cell form the lower roots and other parts, and the suspensor and haustoria. Therefore, this will be very big one, and terminal cell is very small one. Okay. Now, later what happens is, so many divisions happen, anticlinal and periclinal divisions. I am not going to the depth of that because for your syllabus this much is enough. What you have to do, what you will learn is lower cells undergo transverse divisions and form number of, you can see so many elongated cells are formed. These are all forming the suspensor. What is suspensor? The small stalk of embryo that push the embryo into the endosperm to get the nutrients. If there is no suspensor, it remains down and endosperm is above, then it cannot get the nutrition. Therefore, first so many peripheral divisions happen to form the line of the linear sequence of cells called as suspensor, and the basal cell whichever is present, it becomes larger with the larger nucleus that act as a haustoria. Haustoria, the one get nutrition. This is the haustoria. Then upper terminal cells undergo transversely and longitudinal divisions, and they divide into peripheral layer. Actually that will become the dermatogen layer. In future it is called dermatogen, from the epidermis of the layers. Then inner one form the proper embryo structure. Then further, this stage is the starting of the embryo. This still here it is called the pro-embryo stage. This is called the globular embryo structure. So you need to learn the structure. Formation of globular embryo is circular embryo with the basal haustoria and a long suspensor cell. Now this will continue to divide the embryo tissue. Then it will have one small nick-like structure formed in the center, and it buds with two buds of cotyledons. Therefore it looks like a heart-shaped structure. It is a heart-shaped embryo. This is a heart-shaped embryo. And near the tip of it, plumule initials are present that are going to form the shoot tip in the future. And lower cells of this embryo form the radical initials. They form the radical into the root system. Then this is haustoria, this is the suspensor. The whole thing will be pushing the embryo into the deep into the tissue which is endosperm, helps in the nourishment of the embryo. Now at the end of this you get a fully developed dicot embryo. It is a fully developed dicot embryo will be like this. Okay, this will have a haustoria. I have just reversed it, and there is the suspensor, and there is a huge embryo found here. You can see this is the body of the embryo. These are the cotyledons, and this is plumule. This is called the hypocotyle. Lower to the cotyledon there is a hypocotyle area. These are the two cotyledons. This is the importance of the cotyledons. Two cotyledons dicot embryo. Therefore, at the end of the development, the complete seed looks like this. Dicot seed, dicot embryo looks like this. Here is the lower part. This is forming the root cap, and this is the radical. Radical is the root initial structure, and this is called the hypocotyle. Means below the cotyledons there will be hypocotyle, and these are the two cotyledons, and this is the plumule. They will ask only this diagram to you. Draw and label the structure of a dicot embryo. This is very important for you. This you draw, and if they say early embryo is this. Actually this, if you draw it, it's a very simple one, but if they ask early development of dicot embryo, means you have to draw this. Right? This is the early embryo, this is fully developed embryo. This is the early stage. Okay? Now, next is about the monocots. Actually in dicots, most of the nutrient materials will be situated in the cotyledons. But when you talk about the monocotyledons, this will be similar, but there will not be a heart-shaped embryo. It will continue as an elongated embryo. There will be a single cotyledon, and it is pushed away. You can see this is the embryo, single embryo that is pushed away from the major part of the embryo. Embryonic axis is pushed out, and that is called the scutellum. Scutellum is the monocotyledonous embryo. Name of the monocotyledonous embryo is scutellum. The remaining embryonic part contains coleoptile. Coleoptile is the shoot tip, and here is the shoot tip is here. Coleoptile is the covering of the shoot tip. You can see this is the covering of the shoot tip. Coleoptile, and the lower portion is this is the radical, and this is the root cap, and this is coleorhiza. Coleorhiza is the covering of the root cap. So this is the total monocot embryo. This also will come at the examination for draw, label monocot embryo, or draw and label the dicot embryo. Remember, these are two important drawings. But whenever they ask you the early stage of development of dicot embryo, you are supposed to draw this, right? So this overall structure you remember, although it looks like more detailed descriptive, you have to know because it is already given in your NCERT book only, right? I have given you the details, and I expect you to stop this and pause it and or take a screenshot and draw them, because they require little more skill from this. This requires skill to draw, right? So I have drawn load of diagrams on the board because I don't want to just keep on drawing, because lot of small cells I need to draw. Therefore, I didn't waste time. Just you have to stop this and take a drawing sketch, or take a sketch beautifully. You draw all of them and practice them. Okay. So that's about the development of embryo in dicots and monocots. Students, once we are finished with the first part, that is about how embryo develops, now we will move on to the next part. What is the next part? The embryo develop into fruit, right? So fruits are formed after fertilization of the zygote, right? So the fertilized ovary or mature fertilized ovary becomes a fruit. So how do we define fruit? Fruit is a mature fertilized ovary, right? And the seeds are the fertilized ovules. Okay. So now coming to the fruits, you would have learnt in first PUC, but still I will give you the details here. We divide the fruits into two types here according to your second year part. Some are called as the true fruits, some are called as the false fruits, right? So what is the true fruit? The fruit that develops from the embryo is said to be true fruit, right? Under true fruits, again we have types. One is about the dry fruit, and the one is the fleshy fruit. Dry and the fleshy fruits. What are dry fruits? The fruits whose the outer covering, because the pericarp is dry and not edible. This is called as the dry fruit. Fleshy fruit are those whose pericarp is fleshy and edible. For example, dry fruit 1 I will give you. Example, first time I am giving you the legume fruit. The fruit is legume. So biologically I will tell you what is leguminous fruit. The fruit that develops from monocarpillary unilocular ovary, superior ovary, containing ovules on the marginal placentation, but it dehisces through two sutures. That's a definition for the leguminous fruit. So whenever you observe the leguminous fruit, maybe a pea fruit you are observing, pea fruit when you study, it will be somewhat like this, right? Like this. And we have openings from both sides. This side it open, and when it opens, you can see there will be one side, pericarp, mesocarp, endocarp, all of used. This is dry, right? On this, there will be ovules attached to this, which are on marginal placentation. So you can see here I am drawing marginal placentation, placenta it is here, right? Like this. This is the leguminous fruit. In pea or beans it will be like this. Okay, legume in pea. Actually, in pea you call, in specific we call it as a pod also. Pod because having very few seeds is pod. Normal legumes have 8 to 10 seeds, like tur dal and other things. They will have long the leguminous fruit. This is leguminous fruit. It's a dry dehiscent fruit. Okay. So what is dry referring to? Not what you eat is dry fruit. My students will say dry fruit are nothing but your grapes and all are dry. No. Dry fruits are those fruits whose pericarp is a fruit wall. This is the pericarp. Pericarp is dry and not edible, and these are the seeds. Okay, seeds are edible. Okay, now we will move on to the fleshy fruits. What are the fleshy fruits? I will write the false fruit later. Fleshy fruits are those whose fruit wall is thick and fleshy and is edible. For example, I will give you for the fleshy fruit. So I'll give you the example of drupe. Drupe is the name of the fruit. Okay, in drupe of the mango, what you will get is the mango fruit. When you observe, you get the fruit like this. What is this? The outermost layer, whatever you are getting, it is a apical, which is green colored part. This is apical. Middle one is mesocarp and inner one is the endocarp. This is the seed. Okay, this is mesocarp and this is the endocarp. Together we call this as the pericarp. Pericarp is the name of the fruit wall and this is the seed. Okay, so this is about the fleshy fruit of mango. We call this the drupe fruit. Okay, now dry and fleshy both are two fruits now. Then I told you there is a second type of fruit that is called the false fruit. So what is the word false meaning? Here the fruit develops from any part other than the ovary. Means it's not actually ovary. The edible part of the fruit is not the ovary. Therefore we call it as a false fruit. This is seen in the pome of apple. Pome is the name of the fruit, apple is the example. So whenever you observe the apple fruit, whatever you get at the center is the ovary. This part, whatever I am drawing, is the ovary. Okay, and here is the fruit thalamus. This is a thalamus what I am drawing. What we are eating is thalamus. It's not the fruit wall, right? So here are the seeds. You get the seeds over here, and this is the ovary, which normally we will throw it off. That is the ovary with the ovules of the seeds. These are the seeds. But major part, what is edible, this whole thing, whatever reddish part you get here, it is nothing but the fleshy thalamus. Fleshy thalamus. So therefore, it is called the false fruit. Why? Because its edible portion is not the fruit, because we are not going to eat that. Only the thalamus is edible and that becomes fleshy and thick, and that is why we call this apple fruit as a false fruit. It comes under the group pome. Pome is the name of the fruit and example is apple. Okay, remember, distinguish between two fruits and the false fruit. You have to write at least two differences. First is the fruit develops from the mature ripened ovary. Here the fruit develops from other than the ovary. Then here what you will write, under fleshy fruits we have the epicarp, mesocarp, endocarp, or the pericarp is sometimes dried. It is in dry fruits. Sometimes it epicarp, mesocarp, endocarp, one or all of them. That is, a pericarp is edible in fleshy fruits. So here the edible part is the epicarp, mesocarp, endocarp. Everything is edible here. Here the fruit wall or the pericarp is not edible, only the seeds are edible. Therefore it is a dry fruit. Then what is the false fruit? I told you it is the fruit that develops from other than the ovary. Here the fruit develops from the thalamus, which is edible. The ovary and the ovules are not edible. That's why this is a false fruit. So this distinction you should do. Okay? Right. So these two differences you understand. Drawing also may be asked sometimes, or directly the questions may be asked. Right. Read. Once we are finished with the previous thing, we are now going to end with the chapter plant reproduction. That is the last part now I am giving you. That is, after fertilization, once the fruits are formed, that's the end of regular fertilization, regular plant reproduction. But there are some exceptions where you would have seen sometimes the fruits are having seeds, some will not have seeds. Like papaya and all sometimes without seeds. Grapes are seedless, bananas are seedless. Then you'll be wondering, okay, if the plant is undergoing fertilization and producing the fruits and seeds, what is the reason why these particularly fruits don't have seeds? That is common question to all. So that is why the last part what I am giving is some of the unusual things happening in the plant. One is called apomixis. So the word apomixis, I given the meaning here. Apo means without, right? Mixis is mingling, means fruit is formed without undergoing fertilization. Means there is no union of gametes. Like its reverse is amphimixis. Amphimixis, mixing of male and female gametes are happening. That is in fertilization. We already discussed about it. That's a male gametic nucleus, female gametic nucleus fertilized to form the zygote. This is the other word. The opposite word is apomixis. Apo means no fertilization. Without fertilizing or without mixing, there will be formation of fruit. So what are the types in this? This is having three types. Agamospermy, second is adventitious embryony, third is diplospory. Right? For the details I am giving you, otherwise just writing formation of fruit without fertilization is fine. But still you should know details, it is a neat portions. In detail you have to learn that. So agamospermy, that means embryo is formed without meiosis or syngamy. Means from any sperm is formed. Normally spermy means formation of gametes with meiosis. But if it is happening agamospermy is the embryo formation without the regular meiosis or syngamy. Syngamy is the fusion of gametes. Both are not happening. That's why it is called agamospermy. Agamospermy is without the formation of any meiosis, no reductional division, no meiosis happen. They are all diploidally group, right? Next is adventitious embryony, adventive embryony. Adventive embryony means the embryo is formed from the diploid sporophytic mother cells, sporophytic cells. And whenever you observe within the embryo sac, you get so many diploid cells, right? Nucell is diploid, outside there is the nourishing tissue, is a nucellus is diploid, and integuments are diploid. Only within the embryo sac you get all haploid, haploid synergids are there, haploid egg is there, haploid polar nuclei are there, haploid antipodals are there. But outside all are diploid structures only. So that is called adventive embryony. Means embryo is developed from any diploid cell, that is the sporophytic cell, mainly the nucellus or the integuments. Nucellus, I told you earlier when we were talking about the reproduction, the megasporogenesis, from nucellar archaeosporial cells only, the other things will distinct. They will undergo meiosis and form the megaspore mother cell, megaspore mother to megaspore and everything. But I am explaining here the adventive embryo is developed from the diploid sporophytic cell like nucellar cell or the integument cells. Therefore, these are all diploid in nature. All these are diploid. Therefore, embryo is formed in a diploid condition only is required. But they are not undergoing meiosis. They are not undergoing after meiosis. They are whatever they are formed. They are not forming embryo only. They are formed without mixing, without fertilization, without mixing of the male and female nuclei. So this is adventive embryony. Third one is diplospory. Diploid condition. So embryo sac first is developed by the megaspore mother cell. Megaspore mother cell, already I told you, it will undergo linear tetrad, nothing is happening. The complete megaspore mother cell will form the complete embryo sac. It is diploid, right? Then within that unfertilized egg, because already this is already diploid, and unfertilized egg will grow into without parthenocarpy. This embryo sac is diploid, but the egg is haploid, right? This haploid cell, without undergoing fertilization, it will form the embryo. Therefore, it is called as a parthenogenesis. Undergoing no fertilization, egg will form the embryo. This is called the haploid embryo. These are the haploid embryos. So this is N number. Embryo is haploid in nature. This is haploid because parthenogenesis is happening by the egg itself, but egg is not undergoing fertilization. Then last one is under this apospory. What is apospory? Nucellus, which is diploid in nature, it will form an embryo sac, and the diploid egg, because embryo sac is not formed from the megaspore mother cell. Therefore, if the nucellus only is forming the embryo sac, the whole whatever it is producing are diploid. Then all the diploid egg will directly develop into embryo. So therefore I have given you this chart, because you should not confuse, oh it is egg, this is the diploid embryo, this is haploid embryo, right? But apomixis itself means without the union of male and female gametic nuclei, the diploid or haploid embryo is formed. Right? So all the three things you just keep it in mind. Lot of questions appear from this. Remember, in NEET you will get apomixis questions, because always in NEET they will expect you to learn all extra from the normal. Normal up to fertilization is normal, right? Now whatever I am explaining is extra thing from NEET portions. Then comes the parthenocarpy. What is parthenocarpy? It is the formation of the ovary into fruits without fertilization. Means fruits are formed without fertilization. Seeds are formed, no fertilization. Here the seed problem I told you, the embryo formation. The ovary develops into fruits without fertilization. So method how the parthenocarpy happens, if at all the ovary is there, means all meiosis and all would have happened. Then how exactly? So after parthenocarpy, seedless fruits are formed. Right, like grapes are seedless, right? And banana is seedless. How are they formed? That is by parthenocarpy. Very nice thing is, it's like a fooling the flower, right? Normally pollen grain enters, pollen tube forms, then only fertilization happens. What the plant breeders will do, and why they will do it? Because seedless fruits have great economic importance. A lot of people always want to buy seedless grapes rather than seeded grapes. Why? Seeded grapes, they will be not that sweet, because all the nutrients is taken away by the embryo. Therefore they don't like. Therefore the parthenocarpy is taking more importance, because people want everything easy and sweet, and everything should be very tasty. That's why parthenocarpy is induced by plant breeders. How they do it is, they take the pollen grains of some other species and transfer. What happens is, the embryo will feel the pollen grain has already transferred, the pollination has happened. Then after pollination happens, then only the pollen tube should develop, then other things will start. But once the pollination happens, there will be misunderstanding of or the embryo will understand that, or the ovary will feel that already the fertilization has happened, it start producing the endosperm. Therefore the ovule will remain non-fertilized, unfertilized. Then the embryo will have all the nutrients of the endosperm without the seed, without the ovule getting fertilized. Therefore it is like fooling the flower, right? Apart from that, they do one more thing, they kill the pollen grains. Pollen grains, they will kill it, transfer it. So what happens? That will not have any male gametes. That time still the ovary will misunderstand as if but pollination happened, fertilization will happen. So I start producing the endosperm. That's how fertilization is not happened, but still the endosperm is formed, egg remain abortive. Means it is not working, it's abortive ovule we call it. It will not have any functional characters, because this is not at all fertilized. Therefore parthenocarpy takes importance. Then how I told you, use of transfer of dead pollen grains or the different pollen grains, or use of auxins. Already you know that auxins are inducing parthenocarpy. Auxins are some type of gibberellins also. If you transfer or spray the flower with auxins and gibberellins, then the flower will immediately, the embryo will be immediately starting with the formation of the endosperm. No egg is fertilized. Egg will remain abortive. That one of the example is NA. You know, I hope you know this. NA is naphthalene acetic acid, a type of auxin. This is going to induce parthenocarpy. The example is banana, papaya and grapes. Okay, and the last part is polyembryony. Polyembryony is development of more than one embryo within the embryo sac. Already I told you, due to apomixis also it may form. I told you here, agamospore, embryo is without meiosis and syngamy, and advantages you can see nucellus integuments all are developing. Then inside, suppose the real embryo formed also, it will be fighting with these to grow. So many times it may not grow, it may grow. If it grows, nucellus also forms an embryo, integument also forms an embryo, and the Regular embryo also will be formed inside by formation of the zygote developing embryo. All is forming polyembryony. Many embryos are developed. You can see here polyembryony, more than one embryo is in the embryo sac. Many embryos will be there inside. How they are formed? First is called cleavage polyembryony. Cleavage means egg is there, no? Egg will normally divide. Means fertilization happens, zygote is formed. The zygote will start, is not egg, it is actually zygote. So once the zygote begins to develop, it will undergo cleavage. Cleavage, first cleavage, second cleavage, divisions happen. No? All whatever they are divided, all will develop into embryos only. That's called the cleavage polyembryony. It is not forming one embryo, it forms, just now I told you how exactly embryo is formed. It forms one basal cell, one outer cell, but all the cells which are formed, no suspensor, nothing, everything will develop into embryos only. Therefore, it leads to polyembryony, we call it the cleavage polyembryony. Then is false polyembryony. What is false is, it looks like polyembryony. Many embryo sacs and many nucellus are formed. Means embryo sac only there are a lot of nucellus and the embryo sacs are formed within the ovary. All will grow as embryos. It's looking like the polyembryony, but it is actually not polyembryony because embryo sacs are only many, but they look as if it is polyembryony. So the example for cleavage polyembryony and this is citrus fruit. That is, citrus fruits have many embryos within the embryo sac. Okay, so that ends the plant fertilization and plant reproduction. Right? We are going to, after this study, some of the board examination questions, and apart from that, 1 mark, 2 mark and 5 mark questions I will give you, and then its answers. Okay, that is the today's class. After these things, we will discuss all those questions, and also about the plant reproduction, also about the human reproduction. After that, we will start with the reproduction in organisms. Okay, so that ends today's class, and that is ends this not today's class, today's subject, that is plant reproduction. After this, I am going to only explain you about the board questions and need portions, how much you have to concentrate. Okay.